If you need help with understanding membrane transport, this video will cover diffusion, osmosis, facilitated diffusion, endocytosis, and exocytosis. As a quick review from simple cellular anatomy, please remember that the cell membrane is composed of a phospholipid bilayer that contains integral and peripheral proteins, cholesterol, glycolipids, and glycoproteins. There are two different types of membrane transfer that occur: passive transport and active transport. In passive transport, materials cross the cell membrane without the input of energy. In active transport, energy is required in order for materials to cross the cell membrane. Typically, ATP, adenosine triphosphate, is utilized as the energy source. ATP is commonly used to fuel metabolic processes throughout the body. Diffusion is a type of passive transport in which molecules or ions move from an area of higher concentration to an area of lower concentration. The slide displays an example of simple diffusion in a solution. A solution contains two parts, solute and solvent. The solute is the thing that will be dissolved. For our purposes, the small purple molecules. The solvent is the thing that does the dissolving. For our purposes, the green portion within the container. Diffusion will disperse the solute molecules so that they are evenly distributed in the container. Diffusion may occur through the cell membrane. For example, oxygen carried in our blood is constantly being distributed throughout the body. When the concentration of oxygen molecules is greater outside of a cell than inside a cell, oxygen will diffuse into the cell. Osmosis is the diffusion of water through a semipermeable membrane. In osmosis, water moves through the membrane. The solute does not move through the membrane. As the illustration shows, water will move to the compartment that has the higher solute concentration. After osmosis, both compartments will have similar concentrations of solutes. Facilitated diffusion follows the concentration gradient and is the movement of molecules from high concentration to low concentration, just like simple diffusion. However, facilitated diffusion requires a carrier protein to transport the solutes across the cell membrane. As a review from simple cellular anatomy, endocytosis brings extracellular materials into the cell by utilizing a vesicle that is gradually formed from the cell membrane. Endocytosis may be classified as either pinocytosis or phagocytosis. Pinocytosis is utilized to bring in smaller molecules and can be described as cell drinking. Phagocytosis is utilized to bring in larger molecules and can be described as cell eating. The mechanisms of these two types of endocytosis are slightly different. When looking at exocytosis, exocytosis transports intracellular vesicles outside of the cell. Exocytosis often serves to secrete hormones or release neurotransmitters, and also can be used to export waste outside of the cell. When looking at the illustration for pinocytosis, you can see a slight indentation form in the cell membrane, and eventually it completely surrounds the small extracellular molecules. Invagination is the term used to describe the process of pinocytosis. When referring to the illustration for phagocytosis, you can see that a portion of the cell extends itself around the molecule. These cytoplasmic extensions are called pseudopodia. After endocytosis occurs, the vesicle may be broken down and is recycled. Its contents can be digested and utilized as needed by the cell. I hope you found this video helpful. Please feel free to subscribe for additional simplified anatomy videos.